Hello, my name is Kayla Brogan, and today I will be presenting to you my study titled An Evaluation of the Evidence for Ocean Acidification Across the Jurassic Boundary. Did you know that in the past 200 years alone, ocean water has become 30% more acidic, faster than any known change in the ocean chemistry in the last 50 million years? Figure 1 shows the increasing rate of ocean acidification from the time millions of years before the present. So what is ocean acidification? Ocean acidification is associated with low pH levels. It is caused when CO2 is absorbed into the ocean's biotic structure, contributing to the decrease of pH levels. It is also directly linked to climate change. Disturbances in marine ecosystems due to ocean acidification include colonies of coral reef bleaching, rock sediments becoming degraded, and the negative effects of fish populations and invertebrates. Figure 2 shows one of the disturbances to marine ecosystems, or coral bleaching, and Figure 3 shows the trend of CO2 increasing in oceans and pH decreasing. So, my study will focus on paleocean acidification, which is ocean acidification in the geological past, and I'll specifically look at bivalves and rock record and shelves, which are a great record for ocean acidification, and they are a great archive. However, ocean acidification over many years has degraded the rock sediments, making the fossils hidden underneath a deep matrix, and the Jurassic is rich in acidification events. Photo 1 shows an image of 183 million-year-old fossils from my mentor's lab comprised of bivalves and shells, and Photo 2 shows a tool being used in an air pressure machine to remove that deep matrix. So, an event that caused acidification in the Jurassic was the Tortian Oceanic Anoxic event, or the TOWE. It released immense CO2 into the ocean's biotic structure, which completely degraded the rock record and contributed to a cause for ocean acidification in the early Jurassic. Figure 4 above shows the location of the TOWE in geological times. So for my study, I focused on 3 different variables, or characteristics, which included mobility, which is how an organism moves, and the mobility level reflects the spectrum of activities an organism can achieve in response to disturbance and or external stimuli. It was organized with the following measurements. Stationary category, which means the organism was stationary and couldn't move. Unattached facultative moving means that the organism was free to move and was moving. And pedically attached mean it was semi-moving. It was attached, but it could also move around a little or swim around. Unencumbered means it was freely swimming or freely walking. Tiering is the vertical ecological structure present in benthic marine communities. And the levels here are epi and faunal, which means it was buried deeper down in the ocean's core. Semi and faunal is a little higher than that. Shallow and faunal is the surface level of the ocean, and deep and faunal is buried all the way down. Feeding is the means of acquiring energy necessary to maintain life, and here we have a herbivore, which is eating plants and such, and carnivore is meats and other animals or organisms. And there's also suspension feeder, which means that an organism needs help eating. So for my review of literature, Ettinger and Larson in 2020 took 293 samples of carbon and oxygen isotopes of rock sediments, which were analyzed from the Tortian, now located in the Italian Apennine, and they measured both thickness and frequency of the rocks and found that both had decreased since the Tortian. Green and Martindale in 2013 first identified abiotic and biotic responses to ocean acidification, and they used these factors to help identify rock sediment from the Triassic-Jurassic time period and found that they were only slightly affected by acidification. Martindale and Foster in 2022 utilized 2,300 fossil shells from marine snails and bivalves to test for acidification, and they found that marine animals with shells made of aragonite are particularly susceptible to acidification. And for my gap in literature, articles have shown to understand how ocean acidification impacted organisms in the Jurassic, but none have compared the changes of certain species before and after the early Jurassic. So my purpose of study or my research is attempting to figure out if ocean acidification caused a change in the biotic structure of the early Jurassic by looking at fossils of organisms from that time period or the Jurassic and comparing them to modern day organisms, while also taking a brief dive into the Tertian oceanic anoxic event, an event that primarily affected marine life. So for my methodology, first I identified different types of fossils from the early Jurassic from previously collected data. And so out of the 93 organisms that were collected from the Jurassic, they were first separated into categories such as bivalves, gastropods, brachiopods, and solentrates. And then they were further separated into subsections based on their genus and species. And from the number of modern-day organisms, I collected variables such as tearing, mobility, and feeding, and I organized and compared modern-day organisms to Jurassic organisms. And from the number of extinct organisms, I compared different organisms from modern day to extinct. And finally, a chi-square test was used to run the frequency of the two groups. For my results, figure 5 shows a diagram of the number of organisms in each group that were extinct or extinct. And out of the 93 organisms that were collected, only 10 were extant, leaving a 9.6% survival rate since acidification events in the Jurassic. Extinct are the organisms that had died off, and extinct are the ones that are still prevalent today and still around. Figure 5 shows a diagram of the frequency of tearing variables, and you could see epi or epiinfaunal is the most prominent here, or the most occurring. Figure 7 shows a diagram of the frequency of feeding mechanisms, and suspension feeder is the most prominent here. And figure 8 shows a diagram of the frequency of mobility levels, and you can see facultative mobile unattached is the most frequent here. Figure 9 shows the most occurring mobility strategies in both extinct and extant organisms, and the most occurring one in the extinct category is facultative mobile unattached, and that was where they were able to move freely. And for the one that was extant, the one here is bystand, and that means that they were able to move in the slightest. And then after I ran a chi-square analysis test, I found a p-value of 0.09, and this means that there's some confidence that there is a relationship or that mobility was a prominent characteristic in determining if ocean acidification. Figure 10 shows the most occurring feeding mechanisms in both extinct and extant organisms, and you can see here in both the extinct and extinct category, suspension was the most prominent and the one that occurred most frequently. Therefore, the p-value was 0.85, and this means that there's no evidence that there was a relationship. So it really doesn't matter what the organisms are intaking in order to sustain themselves. Finally, figure 11 shows the most occurring tearing variables in extinct and extant organisms, and in the extinct category, epiinfaunal, which was the one that was buried deeper in the ocean's core, was the highest or the most frequency occurring, and S-inf or shallow infaunal was the one, not shallow infaunal, the S-inf is the one that's buried very deep in the ocean's atmosphere, or the biotic structure of the ocean, and you can see here that's the most frequent or the most occurring. And here in tearing, we found a p-value of less than 0.1, which means an extremely strong confidence that there is relationship in tearing. So within the tearing category, epiinfaunal occurred most frequently. Within the feeding mechanism, suspension feeder occurred most frequently. Within the mobility levels, unattached facultative mobile occurred most frequently. And tearing was the biggest variable in influencing the number of extant organisms with a p-value of less than 0.01. Epiinfaunal tearing is the benthic faunal living on the substrate or the hard sea floor. So that's kind of another way to say S-inf or S So that means that both of them were buried deeper in the ocean's core. And the organisms at the bottom of the ocean floor are less affected by CO2 as it dissolves. So CO2 starts very strong and then dissolves as it gets deeper in the ocean. And since these organisms were buried deeper, they were less affected by acidification. So for my discussion and inclusion, studying the past enables you to have a better understanding of the future, and future studies can also use other growth characteristics to measure the acidification of Paleocean sediments. Here are my references, and thank you for listening.